once again, I would like to thank everyone at the NDIPD for this opportunity to, provide this training. I think this is my second training in about a month. I had one last month. It was an exciting time, and I must confess that my experience with that training actually excited me, motivated me. I'd been like, yeah, when is the next one going to happen? Because this is a good initiative. Amanda this morning was talking about health disparities, and I went like, yeah, these are some of the things that are being caused by the issues that I will be talking about in my training this afternoon, and it's about fears. One of the things that one of the factors that drive these disparities, and actually I use the term widen the disparities, is related to the fact that there are so many fears about testing, there are so many fears about vaccines, and so yes, like Martin said, it's going to be quite interesting. And some of the things I'll be talking about this afternoon, they are available there in the public domain, to be honest. But I will be taking my time to explain them and to provide some, reasons behind what is going on or what has happened in terms of testing and in terms of vaccine. So this afternoon, I'm going to split the training into two. So we'll talk about tests first briefly, because I don't think fears about tests is as huge as the fears regarding. Actually, I like to use the term reservations, not necessarily fears. In many situations, the problem is not just about fears. The problem is related to reservations. People have reservations. So we will be addressing that mainly. And yes, I have time to myself, and I expect to finish this training in an hour, 20 minutes, thereabouts. We will be taking a short break after talking about tests. I would want us to take a short break, catch our breath for about 5 minutes before we go on to talk about vaccines. Now, so this I like to do a lot of talking, a lot of description about the things I'm talking about. So why do we need to get tested for COVID-19? This is a situation that came upon us suddenly, I must confess. And it was so sudden that the whole world, including the scientific community, we were all thrown into a very chaotic situation. We did not really understand this virus. And I must say that we still do not understand everything about this virus. And that's why we've got the Delta strain, which hopefully I need to talk about later. But why do we need to get tested for COVID-19? I have been in situations where, someone I know, has a son who was showing symptoms of COVID-19. And, this young person was saying that the way I'm feeling, Mom, I need to get this looked into. I think the symptoms, they are similar to the symptoms of COVID. The mom said, no, an African parent would go like, no, who told you you've got COVID? It's not COVID you've got. And then, there are other spiritual angles, introduced to it, no, it's not your portion and all that. But this young person insisted on getting tested. And eventually he was tested positive for COVID-19. So, every time I think about what might have happened if this boy did not get tested or if he didn't insist on getting tested. But the first thing I want to say this afternoon is that testing saves lives. It does save lives. The reason is because the reason is simply due to the fact that, like any other illness, be it, we are told that some cancers, if they are diagnosed early, the outcome is better than when they are diagnosed late. So it's like any other condition. The earlier it is detected, the better it is. for the patient. So if testing is done early, a definitive diagnosis is made, then treatment can proceed early before any damage is done. Okay, so with COVID-19, I need to explain this. With COVID-19, depending on the individual, the condition can progress to such a situation that irreversible damages are done to different organs in the body. A lot of us have heard about long COVID, and that is simply because the virus has done irreversible damage to many organs in the body, and as a result, the organs are struggling to recover. And so, when testing is done early and treatment is initiated, and I'm sure you're going to ask me what kind of treatment, whatever treatment is available out there, if it is initiated early, it reduces the severity of the condition, it reduces the risk of long-term disability or death. So that's one reason. The other reason is this: we simply limit the spread of the virus. When testing is done early, the chances of infecting others is limited. And the reason I'm emphasizing this is because in our community, especially within the Black community, testing is seen as, oh, what if it's positive? Then, I even understand that back in Nigeria, being positive for COVID-19 is like a stigma. It's not supposed to be that. There are very good reasons to be tested. And I also want to talk about pre-symptomatic people and asymptomatic people. And then we begin to appreciate the fact that testing is very important. Pre-symptomatic people are people who are already infected with the virus. So it is possible that you'll be with a person who is already, infected with this virus, but they are still pre-symptomatic. They're not showing the symptoms yet, but they can infect others. And that's another reason why it's important to get tested, even if it is just voluntary testing. I work in an environment where you're required to do a test every week. And then we have people who are asymptomatic. And I think, especially within the Black community, this is a major challenge. We still struggle with understanding this concept of being asymptomatic. And I think, that state of being asymptomatic is driving a lot of misinformation about the virus. So people go like, I tested, this COVID-19 thing is a scam. I did a test. They told me the test was positive, but I'm fine. I do not have any symptoms. So why? What's all the stress about testing about? What's all this stress about COVID about? If they say I'm positive and then I'm not showing any symptoms, then COVID is not a problem. Ladies and gentlemen, it is a problem. The fact that an individual is asymptomatic doesn't mean that they cannot infect others. Being asymptomatic simply means that because of the immune status of the individual, the virus had not been able to evade the immune system. It had not been able to evade the immune response, which is what should have happened normally. But because we are all different, our health status, we all have different health status. Some of us have, conditions that we are not even aware of. And the outcome of an infection with the virus will depend on our status at that time. So there are people who, come in contact with this virus and they do not show any symptoms. And that is simply because at that point, at that time of coming in contact with the virus, the immune system was able to deal with the virus. The virus was not able to evade the immune system. So we have to think about all this when we think about testing. We may be asymptomatic, but it doesn't mean that we cannot infect others. So at the beginning of the pandemic, I've got a teenage daughter, and I would say to her that, yeah, you are young and all that, but think about the old woman on the bus. Think about the guy who is receiving cancer treatment. You may come across Think about all the people who have obesity, diabetes. These are people with pre-existing conditions that if you come in contact with, yes, you are asymptomatic, but these people may not be as lucky as you are. So we have to think about getting tested beyond ourselves. We have to think about all the people we come across, all the people we may infect. And during the early stages of the pandemic, I would say to myself that if I was asymptomatic and I infected another person who eventually died, that meant that I've got blood on my hands. If I knew that I was supposed to get tested, but I became asymptomatic. I became infected, and I infected another person. And the person eventually died. And it was traced back to me. And I was able to know that, this person who died because I infected them. That wouldn't be a nice thing to think about. So it's important to get tested. Now, the other question a lot of people have asked me is this: are these tests reliable? There is no test on earth, and I'm talking about COVID tests, HIV tests, whatever tests, diagnostic test you want to think about this afternoon. There is no test that is 100% reliable. There are times when a test will be false positive, and there are times when a test will be false negative. So that's why with any test, including COVID-19 tests, they say carry out a confirmatory test. Sometimes they say carry out a follow-up test. So if a test was carried out on a person who is, say, symptomatic for the virus, depending on the situation at the time, the test may come out as being negative, even though the person is showing symptoms. So what may happen is, the doctor or the healthcare professional would say, okay, let's use another method, either as a confirmatory or as a follow-up test, in order to know whether this thing is really what we think it is or it is not. So tests are only relevant to the sample that was collected at that point in time. So the same test may be carried out on a sample that was collected at a different time, and the outcome will be different. I'll give you an example. We do a lot of research in the lab, and the outcomes are usually different. Same experiment, same samples, same procedure, nothing has changed at all, and the outcomes are different. And that's what we call biological variability. There are times when things do not always happen in the same way. And that's why you've got to do them again and again and again. So we shouldn't be concerned about the reliability of COVID-19 tests because we have more than one test out there. And at the moment, there are 3 tests that we can think about when we think about COVID tests. And I'm going to take my time to explain these 3 different tests and what they do. At the moment, the gold standard is the PCR test. Now, what does the PCR test do? The PCR simply detects the genetic material of the virus. The virus is a single, we call it a single strand of an RNA. And you can easily detect its presence using this PCR. PCR is a test that has existed forever. I mean, when I say forever, I mean for a very long time. The truth is this same PCR test has been used in several other tests you may have taken. I mean, maybe, you went to your GP and they said, oh, we need to collect your blood samples. We need to carry out some tests. Trust me, some of those tests were done with PCR. So PCR is not new. It's not a new test. It's a test that has been tested over a long period of time, and it's been trusted to produce accurate results for the presence of any condition. It may be COVID, it may be, other types of infectious diseases. The PCR is able to detect them because what it does is to detect the presence of the genetic material for whatever it is that is responsible for the condition. Now, you will go like, okay, so how does that work? We can even use PCR to test the presence of so many things, about us, because we are also made up of genetic material. So if my everything that constitutes my genes, all my genes, if there is a library, if there is a database that contains all my genes, if someone wants to detect whether, this was Mayo or not, they could do a PCR. As a matter of fact, PCR is even used in forensic science. So when they say, we took a material, DNA material or whatever material, maybe they will say, we used the saliva from this person. It's because, the genetic information is known. The other test, which is more rapid. So before I move on, PCR is used as a diagnostic test to check for active COVID infection, and all it requires is a nasal swab. The antigen test is slightly different in that what it does is it uses, the principle of antigen-antibody reaction. I would explain that. The antibodies, the immune system in our body, they are specific. We have different antibodies that detect the presence of different proteins or genetic materials, including viral materials, including, any protein from a virus. So when a sample is taken from us and that sample contains, the protein for COVID, and the protein is actually the spike protein, which I'm coming to talk about later, this antigen test is able to react with that protein if truly the spike protein for the virus is present in that sample. What the antigen in that kit does is to look for it, search and search. The thing that I always bind with is it in this sample or not? If it is there, it will bind to it and it will detect it. And this is also done using a nasal or nasopharyngeal swab. Some of us may have done this already. However, the caveat with the antigen test is this, and that's why some people say that it's not very reliable. It may not detect an early COVID-19 infection as much as the PCR test. The reason is it takes time for the protein to be made, whereas the RNA is always there, so the PCR will pick the RNA at any time. But the protein for the virus, the protein for the virus may not be detected early, especially when the viral load is very low. And that's why sometimes people say, I took the lateral flow test and it was negative. How come I then took the PCR test and it was positive? It's because during early COVID-19 infection, the levels of the protein is not high enough to be picked up by the antigen test. Then we have a different kind of test. This doesn't tell you whether you have COVID. Rather, it tells you if you've had it before. It's a blood test which tells you if you've been infected with the virus in the past. And all it does is to check for the presence of the antibodies. Let me explain that. Every time we come in contact with the virus, regardless of the outcome, God forbid it's not death. So if we come in contact with the virus, whether we become sick or not, our body will produce antibodies to that virus. Because that's what the immune system does. The immune system will detect and go like, we've got a stranger. We do not recognize whatever it is that is here. So antibodies are made, specific antibodies are made to that virus, and they remain in the blood. They remain in the body. And so if you do the antibody test, the blood samples are taken either from the finger or from the vein. If antibodies to the COVID-19 virus are found in that sample, it means you've been exposed to the virus in the past, whether you were ill or not. However, it's not used in diagnosis of COVID-19 at the time of the test, and it does not tell you that you have COVID at the time the test is done. It tells you what had happened in the past. And some people use it to go like, oh, okay, if I already have antibodies to the virus, maybe I'm okay. Maybe I'll be able to fight the virus in future if and when I become infected. So why did I take my time to explain this? We shouldn't be afraid of taking the test. As a matter of fact, the tests are very informative. They tell us a lot of things in a situation where a 100% cure, if you like, for the virus has not been found. I feel that even with the current state of the pandemic, testing would help us a great deal in understanding, our limitations in terms of what to do and what not to do. For instance, if tests are taken regularly, the issue of, children going back to school or children staying at home will be resolved, because then you are able to identify who is positive for the virus and who is negative for the virus. So I'm expected to take all questions at the end of our interaction. We're going to spend a lot of time on vaccines, and so I'm going to spend the next 1 hour. I believe I've got 1 hour thereabouts to talk about vaccines. But before we go into vaccines, I want us to take a 5-minute break. And if you've got questions from all I've said regarding tests, and if you still have reservations about the different tests, I'm more than happy to take the questions. So you can leave the questions in the chat box. So we'll come back, we reconvene at 1:05. Thank you very much.
Thank you, Doctor. So let's reconvene at 1:05. Thank you. Okay.
Yeah.
Yeah, go on, doctor. It's right about time now.
Thank you very much, Martins. Okay, so I hope we've managed to catch our breath, and because we want to talk about something that I find very interesting. It's a very complicated subject. It's not only complicated; it's also very controversial. And um, ladies and gentlemen, vaccines have been controversial for many years. The controversies, the complications surrounding the issue of vaccines have been with us since the days of polio, or even beyond polio. Um, Before I start explaining um, what vaccines are all about, let me revisit what our bodies, how our bodies function, how the body functions normally. Now, I will use this example to demonstrate, the relationship between our immune system or our immune response and the different things that are out there, viruses, bacteria, fungi, this pathogenic. I'll use the word pathogenic. Pathogenic means disease causing. This disease causing, microorganisms all around us, and they are everywhere. Now, the first thing I'm going to say is this: if I've got a colleague who does microbiology proper, and he did a study on our mobile phone. He did something with our mobile phone, and he discovered the amount of the level of bacterial growth on our mobile phones, and it was amazing. It was like, what? The same thing with our keyboards, our computer keyboards. All these things, they contain pathogenic organisms, disease-causing organisms that normally might have caused a lot of people, serious illnesses, or may have even resulted in death. That is one example. The other example is this. Let me take us back to Nigeria. The professor who introduced me to immunology or immunopharmacology, Professor Paco, is from Delta State, and Urobuman, late now. He was my PhD supervisor. And we did a study in UI staff school. We carried out a study in UI staff school. And this study involved taking blood samples from kids in staff school to analyze, to measure the presence of malaria parasites. I'll tell you what was interesting about this finding. We found out that most of these kids, they were playing around, they were all over the place. They were not ill. They didn't show any malaria symptom, but they had high levels of the parasite in their blood. And he gave it a term at the time. He did say that these kids are semi-immune. Semi-immune means that, the immune system is not able to clear the parasite from their body, but at the same time, the parasite was not able to make them sick. It means that, these kids have come to a level where they and the parasites were just, like coexisting side by side. Fast forward, after my postdoc in Germany, we returned to Nigeria with my daughter, and she was, I think, a year plus. Within a week, she was seriously ill with malaria. Within 1 week, she was seriously, this resulted in her being admitted at UCH. This is the science behind what's happening, the immune response. There are situations where when the body, sometimes when I'm referring to the immune response, I'll be using the body, as a layman's language. When the body's own immune response is able to deal with all these crazy viruses and bacteria all around us, it's like a walk in the park. The reason is this. these immune cells that we have, they have been trained, they have gotten used to these microorganisms, and they are able to fight. Every time we come in contact with these microorganisms, they are like, sentries. They go like, no, we do not want you here, get out. So they destroy these microorganisms. Sometimes, many of us, we go like, I've got a fever. What is the problem? It's a slight fever. It's actually maybe aches and pains. We cannot trace it to anything. What is happening in many of the cases is that the body, the immune cells are fighting something. And by something, I mean microorganisms, pathogenic microorganisms. The unfortunate thing is when they are doing this, they are also producing the same chemicals they produce to kill the virus or the bacteria, is the same chemicals that give us the fever, the feverish feeling, the aches and pains. However, there are times when a microorganism is so lethal that the body's own immune responses need to be trained, need to be told. We need you to, the next time, please, we are asking you that, it's like being given an instruction. When this virus comes in contact with this person, please make sure you do something about it. And that's why we need vaccines. There are times when the immune cells or the immune system needs to be told what to do. Because number one, it may be a virus or another organism that is able to cleverly evade the immune system. So it's there, but the immune system simply goes like, well, I can't see it, I don't see it. Now, the slide I'm showing you now is a very heartbreaking slide, but I like to show it all the same. This is polio. And these are children that were not given the polio vaccine. And we can see what the virus did to these kids. This is a virus that attacks, the part of the body that is responsible for, walking. And that's why you can see that in many cases, it results in permanent damage, paralysis. This is polio. But, when I tell my daughter who is 18 today about polio, she goes like, what's polio? And I shake my head. Meanwhile, I have a cousin who suffered from polio because the mom was, same thing we are experiencing now. The mom was told, no, don't take that vaccine. It's going to do this to your child. Very pretty young lady. She now walks with a limp. Thank God for good physiotherapy. She had polio. So that's why we do need vaccines. There are times when, our bodies, the immune system is a very fantastic part of us. It is the system that helps us to fight, whether we know it or not, whether we realize it or not. And I've given you two examples, especially the one that amazes me the most were those children in staff school in Nigeria. That's the immune system. It's an amazing part of the body that fights for us, and it fights really, really hard sometimes. But there are times when a new thing comes, a new thing that the immune system will eventually, understand, will eventually get familiar with. But this new thing is so lethal that it would have done so much damage first. A lot of us would have heard of this thing that our government was thinking of trying. It's called herd immunity. Yes, herd immunity is true, but at what cost? Herd immunity simply means that we are willing to allow some people to die because there will be casualties until the remaining populations the immune responses of the remaining population goes like, oh, this virus has been around for a very long time. I know it already. So I don't need to be vaccinated. I understand it. But we are also dealing with a very tricky virus, which mutates quickly. And I'll come back to that later. And that's why, herd immunity is not a very good strategy, because the question then becomes who dies and who lives. It means we are going back to Darwin all over again, natural selection. The weak dies, the strong remains. And that is unethical. So that's why we do need vaccines. Well, let me explain a little bit more about COVID vaccines. Now, let me explain further how vaccines work. Vaccines are like, the vehicle, the mechanism that is used to prepare the body for a bad guy. And how do we do that? It's very funny. What we do is that we look for another bad guy that will not harm us. We introduce it to the body. That bad guy, has, it's like, what we call fake currency or fake this or fake that, something that looks like the original, but not the original. So what we do is we introduce something that looks like the actual bad guy. We introduce it into the body. And when we introduce it into the body, we do it in a way that teaches the immune cells, the body's own immune response, to respond when the real bad guy comes along. That is vaccinology is complicated, but the strategy is very simple. The strategy is simply a strategy involving, causing the body's own army to be prepared every time. So the body's own army, the body's own defense system is the immune system. So what vaccines do is to introduce a fake enemy, an enemy that looks like the original enemy, introduce it into the body, and the body simply goes like, so there is an enemy that looks like this. Okay, the next time we are going to see what's going to happen. We are ready. And that's the simple thing. Now, with COVID-19 vaccines, now I'm using plural, there are different strategies that are being adopted to achieve this purpose. And what I want to do this afternoon is to demystify all these strategies. So you see this thing about vaccines is no big deal. It's simple biology. Simple but clever biology. And I'm coming to one strategy that really blew my mind. It is simple biology, but very clever. So we have 4 different strategies at the moment for making COVID-19 vaccines. One is using a viral vector. Let me explain viral vector. A vector is like your car, your bus, anything that can take you from A to B is a vector. Anything that helps to deliver anything from one place to another is actually called a vector. It moves it. Now, what a viral vector does, a viral vector-based vaccine does is, another virus is to use a harmless virus, maybe the cold virus. The cold virus, the one that just gives us the sniff, sniff, sniff. So the cold virus is taken, its genetic information is modified slightly. We all have genetic information, by the way. Its genetic code is modified slightly, and the COVID-19 genetic code is inserted somewhere. So what happens is the virus is injected into the body, and when this virus is injected into the body, our antibodies will recognize the COVID-19 genetic code. Remember, this is a virus that will not harm, but it's a virus that contains just a small part of the COVID-19 genetic code, the critical part that the that the body's own immune system will recognize in order to fight in future when the actual COVID-19 is exposed to the body. So that's one strategy, and that's the viral vector strategy. The second strategy that is being used, and I'm coming to tell you which vaccine is viral vector and which one is which later. But let me explain, the biological basis for these vaccines. The second one, and that is the most mystifying of them all because it's new technology, is the RNA vaccine. Now, ladies and gentlemen, 10 years ago, I started teaching this module at university. We call it gene therapy. That's gene therapy. And every time I give lectures on gene therapy, these are pharmacy students. They go like, really? Is that possible? And we've been talking about gene therapy for many years. And when the RNA vaccine was introduced, I went like, wow. I was telling my students we will be there in 20 years. I didn't know that we will get there so soon. Because the use of RNA strategy has been a textbook thing for many years. We just haven't had the opportunity to introduce it. What is an RNA? Okay. Every individual, including viruses, including whatever biological entity you want to think about, we are made of proteins from top to bottom. The hair, the skin, whatever it is, we are made of proteins. Now, the question is, how are the proteins that make us formed? But before we answer that question, I want to explain something again. We are not only made of proteins, the functions in our body, digestion, breathing, whatever, these are made possible by specific proteins. So we are proteins from top to bottom. Now, the question is, how are proteins made? Either in human beings or in rats, in cats, in dogs, or in viruses. Proteins are made by what we call an RNA. As a matter of fact, this is something we teach as the central dogma of molecular biology. So what's the central dogma? The central dogma is if the body or any system, any biological system is going to make a protein, a DNA sequence is first of all assembled, but because the DNA is so big, it cannot come out of what we call the nucleus of the cell. So the DNA is then, the information on the DNA is then copied onto what we call the messenger RNA. So the messenger RNA takes the information out of the nucleus and starts to make proteins. Because the DNA is so big, it cannot leave the nucleus. The messenger RNA copies the information and uses the information that has been copied to make proteins. My brothers, my sisters, ladies and gentlemen, this is the strategy behind the RNA or the mRNA vaccine. How do they work? Now, the genetic code that makes the virus, this is very clever. Every time I think about this, I go like, wow, this is so simple. How come we've not thought about this for many years? Take a component of the genetic code of the COVID-19 of the virus. It's called the messenger RNA. You just need a segment. The segment for the spike protein, the spike protein is the major component of the virus that the body's immune system recognizes. So take that code, inject it as the mRNA, as a vaccine. Guess what happens? This mRNA gets into the body, and what does it do? It starts to make spike proteins. Because that's what it's expected to do. It goes into the cells and begins to make spike protein because that's what mRNAs do. They make proteins. And when the immune cells, recognize the presence of the spike proteins, they make antibodies. And that's why I'm coming to side effects of vaccines later. Many of us don't understand that. You see, when we get these vaccines, we get the symptoms that are slightly similar to what happens when we have the viral infection. And when I received my vaccine and I was feeling like, I went like, yeah, this vaccine is doing what it's meant to be doing. Yes. So that's what the mRNA vaccine does. That's the whole strategy. The whole virus is, we can take the whole virus and weaken it or inactivate it. We call it attenuated vaccine. Attenuated simply means kill. So with this strategy, you take the actual COVID virus, the actual virus, you kill it. Usually we use heat. You kill it, you put it in a vaccine and expect the immune cells to recognize it. The last strategy is to use a protein subunit. So using a protein subunit and using the RNA achieve the same endpoint, the protein. So with the RNA helps the body to make the protein. However, you can also inject the protein as a vaccine. The end product is the end strategy is to trigger an immune response. You just want the body, you want the immune cells to see that, oh, there is an antigen, there is something that does not belong here that should be here. And, they are prepared. So when the actual virus comes along, then they are able to attack. Now, let me start saying at this stage that the fact that the vaccines will do this doesn't mean that we will not be infected. It simply means that when we are infected, the immune system is able to fight. Let's make progress. So, these are some of the COVID vaccines that are around now. So, the RNA vaccine is the Pfizer and also the Moderna. And you will be surprised why they are the most effective at the moment. That's what the science tells us. It's because it's like, I'm not very familiar with ammunitions, but it's like, the target is so specific, because these mRNAs will make just the protein. They will make just the spike protein. And, it's a new strategy. Now, the good news about mRNA technology is this. Now, people are beginning to go like, if this thing works for COVID, why can't we start introducing mRNAs that will make proteins that are bad in the body, which is what I've been preaching in my lectures for years. It's called gene therapy. Why can't we then, if in cancer or in hypertension, the problem is that one protein is not doing what it's supposed to be doing? Well, let's simply make the body to make new proteins then by, using mRNA technology, and watch this space. It means that the whole face of medicine and medical treatment is about to change forever. So viral vector, I'll tell you something about viral vector and why it was completed quickly. Actually, the people working on the viral vector, the Oxford AstraZeneca, what they were doing was Ebola. They were using that strategy to develop an Ebola vaccine, and then COVID happened. And all they did was to quickly adapt that strategy to COVID-19. So the new Ebola vaccine is based on viral vector. Whole virus is what many of us have been receiving. And, if we've received one vaccine or the other as a baby or as a younger person, these were usually attenuated or inactivated viruses. And that's why, it's been used for whooping cough, rabies, hepatitis, HPV. It's been, it's a strategy that has been in existence for a very long time. Using a whole protein has also been used, and it's been used in the treatment, in the prevention of hepatitis B. So of all these strategies, the only new kids on the block are, vaccines which employ viral vector and vaccines which employ the RNA technology. But I need to repeat this. The end product, the end strategy is the same. The end strategy is this: the body's own immune system must be able to recognize something that is like a relative of the virus itself, so that when the real deal, the actual virus comes along, they are able to go like chop, 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 chop. Cool. So those are the types of vaccines we've got. But I want us to talk about something else. What they contain. This has been a major source of contention. I've heard all kinds of things. It contains, the vaccines contain some magnets, some chips. And every time I hear these things, I just smile. I go like, people do not really understand the regulations and the processes involved in being allowed to administer things to human beings. Let me just say this: before any pharmaceutical company, any doctor, or anyone is allowed to inject or to administer anything into any human being, even if it is one human being, it has to go through rigorous regulatory approvals. Now, it was possible for me to just do a list of what each vaccine contains, but I decided to simply go on the government website to do a screen, to shoot a screen, and I've provided the link of where you can get information. Information is powerful, ladies and gentlemen. Knowledge is very powerful. With information, no one can deceive us. Because with this kind of information, you have evidence, as opposed to speculations for which you do not have any evidence. You just believe it because someone says it and another person says it. Every time you ask for an evidence, there is none. Now, so the AstraZeneca vaccine contains what I told you it contains earlier, a vector encoding the spike protein. So they've used an adenovirus from the chimp, and all they do is to insert the spike protein into it. Now, every drug and every vaccine will have what we call excipients. Excipients are things that are needed to be added to, the pharmaceutical substance to either make them to be more stable, to either make them to be easy to administer, and so on and so forth. We call it formulation technology. So these are the other excipients, L-histidine, and so on and so forth. These are meant to ensure that this vaccine is stable and this vaccine is delivered to the right place in the body. So that's what the AstraZeneca COVID-19 vaccine contains. And same thing with Pfizer. With the Pfizer, it's clear. This has to be transparent. It cannot be hidden. And that's why I provide the link. I'll repeat that. You can find it yourself. So the Pfizer, which is also called Comirnaty COVID vaccine, contains, the mRNA itself and other ingredients. We call them excipients, including sodium chloride, sucrose, water for injections. And these are common excipients used in formulating drugs and vaccines routinely. There is nothing new here. Okay, then finally, Moderna contains all this. I looked through all this and I went like, there's nothing strange here, except the lipid nanoparticles, which is a lipid. I'll tell you what a lipid nanoparticle does. This lipid nanoparticle, when you hear nano, it means small. It's on the nanoscale. What it does is to help to deliver either your drug. It's been used in drug formulations even before COVID-19 vaccines. So these nanoparticles, it's like a delivery system. So your vaccine or your drug is enclosed within that, nanoparticle, that lipid nanoparticle. It's lipid because the cell membrane, the outer surface of our cells contains lipid. And so it will be easy for that lipid to cross into the cells and deliver its content more easily. Every other thing, routine formulation products. Okay, so I want us to look at this information about vaccines, and I want us to come to our own conclusions, and I'm going to break this down for everyone. About 2 weeks ago, when the lockdown was eased, I was apprehensive. I was worried. Because, I work in a university town, and the pubs were suddenly open. Everywhere was just tim. And I even hear that in London, I saw pictures, and I went like, we are in trouble. We are in trouble. We are in trouble. I was worried when I saw people mingling, no face masks and everything. I went like, okay, we're going for another wave. And then I started to count the weeks. Usually we know what happens, and then truly the numbers started to go up. Truly, when the lockdown was eased, the numbers started to inch up again. And I said to my wife, we have to be very careful. We have to. And then just around, so the final, July 19th, so it's close to a month now. Usually you need a lag time of 3 weeks for, the numbers to start climbing. And then at around last week, I said to my wife, I said, have you noticed something? Have you noticed that even though the numbers have been climbing, we have not seen the level of deaths we saw during the first and the second waves? But, I'm a scientist. It's all about facts. If it's not, I need to see the evidence. So I want us to crunch these numbers together and come to your own conclusions about the effectiveness of vaccines. On the 18th of December, 2020, I want us to first of all, I want us to compare December 2020, that's 18th of December, 2020, when the cases were 28,507, and 7th of August, that was last week, when the numbers were 28,612. Now, in December 2020, the number of deaths was 586. And it's just that I do not want to bore you with numbers. Then, as we started to approach Christmas, we were approaching 1,000 cases a day with the same kind of numbers, that is 28,000, 30,000. But last week, we had, on the 7th of August, we had 28,000 cases and just 86 deaths. This morning, I think, I've not looked at the actual numbers. I like dealing with actual numbers. I think the numbers were, for the last 24 hours, the number of cases was about 30-something thousand, but the number of deaths still remain at 100 plus. Now, so this was December 2020, 28,000 cases, 586 deaths, representing 2.1% roughly. I did the math myself. And then January, which was not a very nice period in this country, we had 35,000 deaths, which was similar to some of the numbers we've been having on, off in the last 1 or 2 weeks. And at that time, when we had 35,000 cases, the number of deaths on that same day, 28th of January, 2021, was 1,376. Now, it's obvious that something is going on in August. As a scientist, that tells me all that I need, because when I carry out an experiment in the lab and I have this outcome, we say the drug or whatever intervention, in pharmaceutical and pharmacological circles, we call it efficacy. Then we go looks like, in English language, we say it is working. It's not perfect, but it is working because we can see the relative numbers. So as a scientist, I would say that yes, the vaccines are effective. They are not perfect. They are not 100% effective, but they've taken us from 1,376 deaths per day to less than 100 per day. That's conservative. You could actually say to less than 150 per day. Now, if we want to argue that, well, it's not the vaccines, then what? What is responsible? Was it a coincidence that, these numbers, cases and deaths, they are happening like this, just because they are happening like this? It's because the world is getting vaccinated. This is not just happening. Now, the other worry I had was the Delta variant. I was thinking that, okay, maybe the vaccines, because the vaccines were designed to recognize the Alpha variant, and I do not have the time to go into variants, but this tells us that even with the variants, with the new variants, yes, there will be cases when people will be vaccinated and the vaccines didn't work. Of course, they didn't tell us, and nothing worked. They didn't tell us they were going to work 100%. And let me also break this news to you. I work in drug discovery and development. There hasn't been a single drug that works 100%. Not in the history of drug development. There will still be people who will take a drug and the drug will not work. Okay, so I have roughly 20 minutes to go. Let's move on. So that's effectiveness, but are they safe? Now, I must say this. I have worked both in industry, in drug development, drug discovery and development, and I've worked in university drug discovery. I've worked in the two areas. And I will tell you that one of the reasons I left pharmaceutical industry was simply because the work is just too. It's not like I'm tired of hard work, but it's a very strict environment. It is just too strict. The reason it is strict is this: the regulatory authorities are hard to please. They are very difficult to please. So that's one of the reasons I returned to academic research, because then, if I do research and you don't accept it, at least I've done the research. I have the freedom to do the research. But with pharmaceutical companies, they've invested money. And, that time, the 2 years or thereabouts I was in the industry, it was every morning, everyone was so stressed up because you do not know whether the drug you were working on, whether it was going to be approved by the FDA or the MHRA. It was so tense. I went like, no, me, I can't do this. I would rather do research that whether the drug I'm working on is approved or not, I've done it. So with the COVID-19, it was not any different. The regulations to check for efficacy, safety, they were strict. I'll tell you the reason. If a pharmaceutical company says that this thing does not produce or this thing is safe, or we are saying that it's absolutely safe, and it does something else, they will, they can be taken to court and they will lose a lot of money. They may also lose their license, to develop new drugs. So, and with the issue of safety, I'll come to that when we come to talk about side effects. But the other question that people have been asking is this: why would I take a vaccine that was developed in a record time? They want to use me for guinea pig. Someone actually told me that you guys, you just want to use us as guinea pigs. I was so hurt. I went like, guinea pig, you don't have a clue what goes on in the lab, do you? Now, yes, it was a genuine concern. It was a genuine reservation. If you think, how come? Normally this thing would take 10 years. How come it took 2 years? I'll tell you why. For the first time, and even I saw it, for the first time in the history of drug development, people came together. Scientists, everyone came together. I'll tell you what normally happens in science. If I'm doing a research on a new drug, I hide it from other people. As a matter of fact, when I go to a conference to do a presentation about the drug, I use a code because I do not want anyone to steal the technology or whatever it is from me. But for the first time, everyone had to open their lab books to go like, let's come together. So for the first time, there was unprecedented collaboration. People, there was no, did you know that there were in the past, there were pharmaceutical companies that would hide their technology for years. They would just be doing the research quietly for years. And these are some of the things that contribute to the lengthy process of drug development. People think, oh, normally a drug takes 10 years. It's because there are things that were introduced artificially. The other thing that was an artificial barrier to drug development was money. If a pharmaceutical company knows that this thing is not going to make us money, even if it's going to save lives, they won't do it because they are in business to make money. They will not do it. But for the first time, governments of the world had to give pharmaceutical companies money. That is unprecedented, or that was unprecedented. At the moment, funding for COVID stands at least £2 billion because the world was losing £375 billion every day, every month because of COVID. So it's like, let's go for broke. I can also tell you that at the beginning of the pandemic, the UK government simply brought out, UK government that we will ask for money for research and they'll be taking us through all kinds of hoops. We will have to jump several hoops. They simply brought out money. They said, whatever research you want to do, just do. Just do. We want information. Just do it. Just do it. We want information. And that's why I'm not surprised. And of course, many countries opened up additional manufacturing capacity, which is a very huge thing. We were talking about, large populations, you have to produce large doses of the vaccine, and we had that additional manufacturing capacities. Let me show you something. So on my left, you have the normal 5 stages of vaccine development, and then you have what happened with COVID. So these things were not hidden. With the normal stage, it takes 2 to 5 years to discover. With COVID, it was just a few months. And one of the reasons is because the people in Oxford were already working on something similar. They were already working on Ebola. They just had to, they had to repurpose that research for COVID. Then we have what we call preclinical studies, which takes almost 2 years. In this case, it took a few months or it was skipped. See, so they were not hiding anything. This stage was skipped. And what's preclinical? Testing on animals. And that's why at a time, AstraZeneca was busy recruiting volunteers. They were recruiting volunteers that would, volunteer to take these vaccines. So that was skipped. There are other stages that were skipped. And the one I want you to please pay attention to is the regulatory review and approval. 2 years. People had to be called from their holidays. There was, the bureaucracies involved in approval, they were reduced because, number one, people were dying. Number two, the world was losing money. So yes, it was quick, but it was just like if you and I want to go to the hospital in our car, it will take longer. But if an ambulance with the blue lights flashing is going to the hospital or going somewhere, you will see that, the roads will be open. And that was exactly what happened with COVID. All the roadblocks were taken away so that this thing will happen quickly. The roadblocks were put there to make sure that people did the right thing. But now that people had decided to come together, it was easy to take out these roadblocks. Now, let's talk about safety, side effect. I need to say this, there is no safe drug, there is no absolutely safe vaccine. Remember, we are introducing something to the body. And so the body is an unpredictable place. It's an unpredictable machine. There will be one side effect or the other. There'll be one safety issue or the other. So we need to understand that. So with the vaccines, what should I expect? Mild, short term, not lasting more than a week, sore arm, headache, feeling achy, being sick, feeling tired. And you will wonder, why do I have the side effects? It's simply because that's the same thing that happens when the immune system encounters the virus. It's exactly the same. Now, and this is what I said earlier, there have been reports of extremely rare cases of blood clots with the AstraZeneca vaccine. And that, you see, this is what I like about science. If it is happening, nobody will hide it because it's out there. And this has been linked to several factors. And you could see how quickly people went back to go like, so what was wrong and what was wrong? And now we know that it's because of situations arising from levels of blood clotting in different individuals. And quickly they did say, okay, if you are a younger person, it means you are more predisposed to, the side effects of this vaccine. But the truth is, even with the clots, the benefits outweigh the risks. The benefits still outweigh the risks. If the benefits do not outweigh the risks, the vaccine would have been pulled. So what about long-term side effects? There is a scheme that has existed several years before COVID. It's called the yellow card scheme. The yellow card scheme simply is there so that if there is any safety issue or any side effect that was not picked up, even with drugs that were developed over 20 years, people still reported things on the yellow card scheme. Even with vaccines that were developed over 20 years, people still had, issues, side effects, safety issues that they reported on the yellow card scheme. The yellow card scheme is there as the last phase of drug development. We call it phase 4. We call it post-marketing surveillance. So because things were tested on a few people, now we've introduced it into the general population where, you tried something on maybe 10,000 people. Now you are introducing it into a population of 5 billion people. Of course, we will now have issues, and that's what the yellow card scheme is there for. The yellow card scheme is there, and that tells you one thing. It tells you that, picking up safety issues after a drug or a vaccine has been introduced to the market was not a new thing. It's happened in the past, it will always happen. And that's why I told you earlier, there is no 100% absolutely safe drug or absolutely safe vaccine. All we have to do is, if we experience anything that has not been reported or shown to be associated with the vaccine, we go onto that page, we report it, they pick it up and they monitor it. So that's the thing about long-term side effects. And I'm going to end with some of the things I've seen. The COVID vaccines affect fertility. There is no such evidence. I always tell people, show me the evidence. Which study was done to show that it affects fertility or it prevents people from becoming pregnant? If you are concerned, all you need to do is to search. Not search on those funny websites. The government has a dedicated website where you can get this information. So if you need information on women of childbearing age, pregnancy, breastfeeding, and you see the same information, it applies to all drugs. Most drugs, they will tell you if you are breastfeeding or you are pregnant, don't take it. It's very simple. Okay. This one is the one that makes me laugh the most. And it makes me laugh maybe because I'm a biologist. Biology simply doesn't work like that. mRNA is meant to make a protein. I've explained that. It's meant to make the spike protein, which the body's own immune system will recognize. And all the strategy does is to teach the body how to protect against future infections. the mRNA does not go back into the nucleus. That will be anti-nature, if you like. mRNA always leaves the nucleus. It does not have the capacity to go back into the nucleus. And the DNA is too big to leave the nucleus. That's why the mRNA has to copy the information from the DNA when a new person is being formed. The mRNA copies the information from the DNA because the DNA cannot leave the nucleus and brings it out. The DNA contains our genetic material. So, it will alter my gene, I'll become a zombie and all that. I go like, it simply means you don't understand the biology. And eventually the mRNA is broken down once the protein is made. That's what happens in nature. These vaccines are just being made to copy what would have happened in the body naturally. Okay, the last one is it contains a tracking device. Ladies and gentlemen, there are easier ways to track your movement. There are easier ways. It's easy. As a matter of fact, it is happening already. When you go on the website, it says you have tracking cookies and all that. What do you think those cookies are doing? They don't need a vaccine. That's too expensive. That's too cumbersome. That's too, how do I put it? That's high, it's high tech because it's difficult. When you put a tracking device in a biological system, the outcome is unpredictable. It's easier with your phone. They'll say all this location and stuff, they are easy to track what you do, where you go, sometimes what you say. So your bank cards are, they are there with chips to track you anyway. No chips have been found in vaccines, and no vaccine contains any material that makes it possible for any electromagnetic information to be collected. Ladies and gentlemen, I want to say thank you very much. I've got so much to say. It's a subject I'm passionate about because I'm Black and because in terms of health disparities, we are the ones that when we talk about vaccine hesitancy, it's highest, amongst us. And I always go like, when the first figures were released about the number of people vaccinated and their ethnic groups, I went like, oh, why would it be us being hesitant? Is that, at a point I went like, is there something that we Black people know that other ethnic groups do not know? Maybe we have some knowledge, some superior kind of knowledge. But I've come to terms with the fact that it's because we are naturally not very trusting. And I understand why we will be naturally not trusting of new things. So thank you very much, everyone. And I will spend the remaining time to take questions.